Meiosis and genetic variation, AQA Biology. Key terms. Meiosis produces four daughter cells, each with half the number of chromosomes as the parent cell. Diploid, two full sets of chromosomes. Haploid, one set of chromosomes. Chiasmata, the point where two homologous non-sister chromatids exchange genetic material during chromosome crossing over during meiosis. Homologous pair, one set of one maternal chromosomes and one parental chromosome paired up during meiosis. Chromatid, one copy of duplicated chromosomes which generally join by another by a centromere for nuclear division. Genetic variation, which is variation in alleles. Crossing over, which accounts for the genetic variations as genes are swapped. Independent assortment, which leads to genetic diversity as chromosomes are arranged randomly. And locus, the position of a gene on a chromosome or DNA molecule. Allele is one of the different forms of a particular gene, and life cycles show meiosis in organisms. It produces four daughter cells, half the number of chromosomes as the parent cell or plant, wherever it is. Meiosis. So this is basically what meiosis is, and you have 8 stages instead of 4, like you do in mitosis. And to be honest, they're just the same as mitosis, if I'm being completely honest. So you have prophase, metaphase, anaphase, and telophase. And then you have, but the only difference is you have prophase 1 and prophase 2, then metaphase 1 and metaphase 2, and so on, so on. Now, the process of mitosis, you don't really have, like, you need to know it, but you don't really need to know it because they don't really ask you questions about that. They more so ask you about variation, but I'm going to go through it quickly. So, prophase, you have the splitting of, you have centrioles moving to the opposite ends of the cell, and spindle fibers develop. Metaphase, they line up along the equator of the cell, and spindle fibers arrange themselves along the equator. Anaphase, the sister chromatids are pulled to opposite poles of the spindle fibers, and chromatids appear V-shaped. And then telophase, cytokinesis occurs. I mean, that's basically done twice. So the function of meiosis, they form haploid cells with one set of chromosomes, and they rearrange chromosomes with another, which creates variation amongst the chromosomes. So the importance of genetic variation, well, it allows species to adapt to the surroundings through evolution. And what are the 3 types of genetic variations? Well, you have crossing over, random fertilization, and independent assortment. So this is crossing over. So as you can see, you have your 2 homologous chromosomes, and they're next to each other. So you can see the crossing over occurs here, and then you can see a part of this part of blue has been swapped with this part of red, but they're swapped in equal quantities. That's the one thing you have to know. Random fertilization. You have so many sperm cells. And I'm gonna draw 3 because I'm not gonna take up time drawing sperm cells. And you only have 1 egg. Let's do, I don't know, let's do an egg in the color blue. This is not blue. Blue. So you have your egg cell. And we don't know which one, this one, or this one is going to fertilize that egg. They all hold slightly different genetic information. Well, they all hold the same genetic information, but they hold different alleles. So when they fertilize, say this one gets in, this one might have the combination of, I don't know, blue eyes. So this one will code for blue eyes because this already has a blue eyes gene as well. So they will create a child with blue eyes. However, because this one is brown eyes and it's a dominant gene, if this one was to fertilize the egg, then it will create a child with brown eyes. If this one was green, then it could create a child with green eyes. So we don't know which one's going to fertilize the egg. And then independent assortment. When the chromosomes are lining up along the equator, we don't know. It can line up in any order, to be honest. So when they're separated, when they're separated, okay, how am I going to draw separation? Yeah, okay, when they're separated like this, we've got these and then this. Oops. Let's draw a different color for this one. And then this side. So because of the random assortment on which they line up during metaphase, it will result in different things, different characteristics, different genetic variations of cells that are produced. So where do these things occur? Well, independent assortment occurs in metaphase as they line up along the equator, and this can provide any random combinations. Crossing over occurs in prophase 1 as the chromosomes join sections are swapped over and in equal amounts and alleles become mixed. And random fertilization are just when the gametes fuse. So this is not part of that, to be honest, but yeah. So if you like this video, then please like and subscribe. And the next video will be about genetic diversity and adaptation, as well as the types of selections. So I'll see you on the next one.